Hi guys, welcome to Zell Topics. So when I'm reading and explaining things and pointing out the important things, you may please take out your NCRT textbook and read and understand with me. Okay. So we're going with stamen, microsporangium and pollen grains. Figure 1.2a. As you can see, it shows the two parts of a typical stamen, and this is the stamen. Okay, it's a two parts shown over there, and the long and the slender stalk called the filament. So you can see the long and the slender is called the filament, and the terminal, generally bilobed structure is called the anther. So you need to remember that word, that is bilobed, which is our anther. And the proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. So there's a memory. Like I'm a 12th pass student, and when I was in practical, as I studied CBSE, and when doing practical, I was the student who done everything correctly, and at the first I completed all at the first. I'm the first student who completed and just get out of the room with confidence. And the Viva I attended so easily because I read all the chapters, and it was related only to this one chapter and this one topic. Okay, it was so simple. It just asks the things like what we do. So you need to remember things like that. How you do and what are the things you use. It's simple. Okay. And going to the paragraph. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are dithecous, and you need to remember that is bilobed and dithecous. That's important. You need to underline, you need to highlight it, and you need to remember it. Okay, it can be used in viva too. Okay, often a longitudinal groove runs lengthwise separating the theca. As you can see, a red line that separates theca, that is a dithecous. Okay, let us organization. Let us understand the various types of tissues and the organization in the transverse section of an anther. In the 1.3 figure, it's in the next page. We can just see that transverse section, and the bilobed nature of an anther is very distinct in the transverse section of an anther. The anther is four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. As we know, it does have a bilobed and dithecous, and two important points as we know. And next is one important thing is its structure. How is it? Tetragonal structure is four-sided. So it consists of four microsporangia located at the corners, two in each lobe. As you can see, in each lobe, microsporangia is there. Okay, so you need to remember that it is tetragonal structure. The microsporangia develop further and become pollen sac. Okay, that we can see the pollen sac is shown in the text, which was before it was a microsporangia, which is developed into pollen sac. So each lobe in thecas, like in one theca, I guess so. In one theca, it just have two lobes, right? So it's, you can understand from the diagram and read. So that's why I said look into the text so you can understand easily. Then structure of microsporangium. Oh, before that, they extend longitudinally or go through the length of an anther and are packed with pollen grains. This pollen grains. Okay, and it is packed with what? Pollen grains. You need to remember that too. Pollen grains. You need to remember the structure, where it is labeled. Things like that is important. Okay. And structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers. Okay, this is very important as in 12th class you can it is even asked in long answer type, maybe short answer, 1 mark questions. Phillips match the following. It's very important. The four layers. Okay, structure. We're going with the structure. It is generally surrounded by four layers: the epidermis, the endothelium, middle layer, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. So you need to remember the outer three wall layers, that is the epidermis, endothelium, middle layer. It is also asked which layers and what is the function. Function is always asked. You need to remember that. Okay, even in Viva or something in the class 12 board exam or exams, it can be asked for real. It's asked anytime. Even in mass the following long answer Phillips is asked. And the next is very important. That is the outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of a tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissues occupies the center of each microsporangium. Okay, so when the anther is young, we know that a compactly arranged homogeneous cells, it does have a homogeneous cells, that are the same cells called sporogenous tissue occupies the center of each microsporangium. Okay, as you can see in the next page, the sporogenous tissue is occupied in each center. Okay, it's in the young anther. Next is the microsporogenesis. As the anther develop, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrads. As you can see in the diagram, next page, the sporogenous tissue undergo meiotic division to form microspore tetrads. You need to remember that. What it forms? Microspore tetrads. Highlighted. It is very, very important. Okay, and as each cell of the sporogenous tissue is capable of giving rise to microspore tetrad, each one is potential pollen or microspore mother cell. The process of formation of microspore in a pollen mother cell through meiosis is called microsporogenesis. So you need to remember that each one is a potential pollen, okay, or microspore mother cell. The process of formation of microspores in a pollen mother cell through meiosis is called microsporogenesis. Okay, highlighted. The microspore, as they are formed, are arranged in a cluster of four walls. The microspore tetrad, as the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. As we seen in the diagram, we understood that the microsporangia will develop into pollen grains, as pollen sacs, as we see, and in that pollen grains are packed. Okay, and this happens in the way of microsporogenesis. Last is the production. The last product is developed is called the pollen grains. Okay, next, the pollen grains represent the male gametophyte. So it is the male gametophyte. You need to remember that. Next, we also said about the female gametophyte, but remember, male gametophyte is a pollen grain. So in next part, we'll be talking about the pollen grain. Before that, you need to understand the diagrams, and when you read each line, look in the diagrams and remember, and make out small points. Write in Malayalam or whatever languages you know, Malayalam or Hindi or whatever, in your own words. It's fine and you will get it. It's simple. Okay, thank you.